Good morning, ladies and gentlemen. If I may kindly ask you to join your seats, please. We will be starting the session in a few seconds. So, good morning, everyone. Ladies and gentlemen, distinguished guests and colleagues. Good morning and welcome to this session on emerging chemical hazards and risks. Allow me first to introduce myself. My name is Amin Kassouf, and I have the privilege of serving as the Scientific Director of the Global Food Regulatory Science Society G-Force, which is co-organizing this session in partnership with the Dubai International Food Safety Conference for this year. It is a pleasure to be with you today as we explore the changing landscape of food chemical safety, a field that is evolving rapidly under the influence of innovation, shifting consumer expectations, environmental pressures, and evolving public policies. In recent discussions, especially those surrounding what is now referred to as ultra-processed foods, for example, we've seen food additives and processing aids increasingly discussed within the broader context of nutrition and public health. While these debates reflect legitimate societal concerns, they also run the risk of overshadowing the essential and often beneficial role that chemicals and food processes play in ensuring food safety, preservation, and the product's quality when they are used appropriately and based on science. Meanwhile, the emergence of new chemical challenges driven by pollution, climate change, and changes in microbial and contaminant profiles calls for adaptive, science-based regulatory approaches. As we navigate these complexities, it is critical that our decision remain grounded in robust evidence and transparent methodologies. Today, through a series of expert presentations and practical case studies, we will explore how food regulatory science and risk assessment frameworks are evolving to address these multidimensional challenges that we are currently facing nowadays. We will discuss the drivers of change in chemical food safety, review differences in risk assessment approaches across jurisdictions, examine emerging issues such as emerging chemical contaminants, and highlight the importance of weight of evidence approaches in regulatory decision-making. By the end of this session, we hope to leave you with a deeper understanding of the interplay between science, policy, and public perception in the field of food chemical safety, a reinforced commitment to science-based decision-making and proper risk communication, concrete recommendations for strengthening adaptive and transparent regulatory frameworks around the world. So, before we start with our first presentation and our first speaker, let's watch together a first video for this session. Great. So, are we ready to start our session? So now, with that, it is my honor to introduce our first speaker, Mr. Mark Feely. Mark is a veteran in food chemical toxicology with over 25 years of experience at Health Canada, including a decade as Associate Director of the Bureau of Chemical Safety. He now is continuing also to contribute as a consultant in food chemical risk assessment. Mark has actively engaged also in international food safety, serving as head of the Canadian delegation to the Codex Committee on Contaminants in Food, CCCF, and as a member of both the JECFA expert roster and WHO's expert advisory panel on food safety. He will address chemical hazards arising from recycled plastic, a timely issue with global implications for both sustainability and chemical safety. Mr. Mark Feely, the floor is yours.
Well, thank you for that kind introduction, Amin. I mean, normally when I hear the word veteran, I indicate that that's probably old, but anyways, first of all, I'd like to thank the Global Food Regulatory Science Society for providing me the opportunity to speak on this topic. And in addition, thanks would go out to the Dubai International Food Safety Conference for assisting an organization or sponsoring this event. So my talk today will give you a brief introduction to recycled plastics and some of the issues that from a chemical risk assessment perspective will have to be taken into consideration through the continued hopeful use and introduction of recycled plastics. So essentially, I will provide a brief introduction to plastics. Secondly, we'll look at what are the main market drivers for production of recycled plastics. And then thirdly, and more importantly, it's the specific risk assessment considerations that would have to be used when you're looking at recycled plastics in food contact materials. So it's recycled plastics, and I think this is probably not news to anybody, but there's been incredible increases in the use of plastics over the last 50 years. And there's various organizations, both UNEP and OECD, who have looked at projections of the amount of plastics that are currently being produced, will be produced, and more importantly, which is being mismanaged in terms of loss to the environment. So, we see from these figures that only 14% of plastic packaging is collected for recycling. And actually, what 14%, 5% of that is additionally lost to the environment. So really, you're talking about there's approximately only 9% of plastic packaging is currently being collected for recycling. So the majority is not being recovered, landfilling, and other avenues of escaping the collection system are the main ultimate fate for plastic packaging materials. And this is in comparison to something like paper and steel, where the recycling rates are generally very high. So one of the main issues is that what happens to all this plastic packaging material, especially if it's not being effectively recycled or contained. And for the majority of cases, it's leaking back into the environment. And primarily what we see is massive amounts of plastic showing up in the oceans. So, and these are some of the figures that have been taken from the foundation that looks at the new plastics economy. And this is a few years out of date, but essentially it provides you, some estimation that equivalent to dumping the contents of one garbage truck full of plastics into the ocean every minute. Some of the other estimations that have been provided by this organization that actually within a few years there have indicated that the amount of plastic in the ocean will equal the amount of fish. And then by 2050, the amount of plastic will exceed the total amount of fish that's available for collection in the ocean. So, I mean, this is not to say that plastic does not play an integral part in preventing food waste. And, you know, essentially food waste, you're looking at some estimations are that approximately 17% of the world's annual food production is wasted through various loss mechanisms. And more importantly, this wasted food is contributing significantly to greenhouse gas emissions. So recycled plastics, once again, these are just some figures, and the fact that usually what you see is very limited amounts of plastics that are being collected, either primarily from post-consumer use for recycling. And why? So why is there such low rates of recycling? Well, the three main elements are there, and I think they primarily relate to the fact that the cost for recycling plastic is still greater than virgin plastic being created from natural gas, oil, and other non-renewable resources. The other issue relates to principles of circular economy, where plastic packaging is not being produced in order to facilitate recycling. And some of the main types of plastic food contact materials or plastic packaging end up being very difficult to recycle, so the only outcome is actually collection, then forwarding to either waste recovery or just dumping in landfill sites. So the estimates as of 2015, and these are obviously out of date, is that approximately 75% of the 8.3 billion tons of plastic that are produced worldwide has ended up in waste, and only about 9% which that has been subjected to any sort of recycling. So, and there's two main types of plastic, and this is the polyethylene terephthalate, that would be the little portable water bottles that everybody uses a single occasion, and high-density polyethylene, which is a very chemically resistant structure which is amenable to being used in all different types of food. So this is just additionally to follow up on the TOMRA, which is a company that does a lot of recycling and produces machinery capable of sorting and collection for recycling, has suggested that only 2% of the planet's annual plastic production is reused for the same or similar products. And that was the focus back in the 1960s, when food packaging primarily was not made from plastic. It was that economically it was more feasible to recycle plastic materials or food contact materials into use in the exact same types of food. So, and this is just figures that illustrates the fact that, you know, obviously the gray is not what you like to see, because that essentially makes landfill. These are figures that EPA has put together over the year 1960 to 2018. So the gray being the fact that that's landfill or plastic waste, which is mismanaged into landfill. Composted, basically, we'll briefly mention the fact that a lot of the packaging materials, once again, in direct sort of ignorance of the circular economy, cannot be effectively composted. And the very small amount of the dark blue is what exactly is recycled. So in this case, in the US, and these figures will sort of change on a yearly basis, you're looking at almost 80% being landfilled and only about 10% that's being recycled. So these are just, once again, illustrating some of the figures. That provides the EU total, Germany, and the world total. Germany sort of stands out because they have a very effective waste collection system for incineration. So the waste goes directly into, because if you consider the feedstock is non-renewable natural resources such as oil and natural gas. So the energy recovery of these type of plastic materials is very high. And so Germany takes a lot of its municipal solid waste, specifically the plastic, and uses it for energy recovery. So, and this just basically illustrates, that globally only about 9% of plastic waste is being recycled. But more importantly, when you look at the orange, that's the amount of plastic waste that's being mismanaged. So it's lost to the collection system, and generally it's into the environment in uncontrolled dumping sites. So essentially when you look at this, you say the orange and the dark blue are actually bad, and the light bluish purple to, or the light blue to the turquoise are actually good. So there's not a lot of difference between the OECD countries and the world in the fact that you're only looking at of the most, there's a fairly significant amount of plastic waste that's being mismanaged, and worldwide it only ends up being less than 10 or 12% collectively that's being recycled, and this tends to be dominated by only a certain types of packaging material. So when we look at the situation in the United Arab Emirates, it's really no different from the rest of the world. Their future direction is to significantly decrease landfill, increase recycling rates, but also to increase energy recovery. But currently, and you see the production paths are very similar to what we've already seen, is that you have uncollected waste, which is mismanaged. You have collective waste, which has leakage, and therefore becomes mismanaged waste. And then you have the options of either energy recovery, recycling, or landfill. So, and one of the issues in the United Arab Emirates is about 90% of their plastic waste currently tends to be landfilled. And part of that production is plastic water bottles. It's been estimated that the UAE on an annual basis uses approximately 4 billion plastic water bottles. So if you think that you have 4 billion plastic water bottles and 90% of those end up as mismanaged waste. This may appear to be bad news, but actually we will revert back to a good news story for UAE near the end. So what happens to the plastic waste? Well, 80% of the plastic waste that is landfilled ends up in the ocean. And there's enormous amounts of waste that's transported by rivers into the ocean. This is just one example. It's called the Great Pacific Garbage Patch. It's about 3 times the size of France, and currently has anticipated to consist of almost 129 metric tons of waste. And of that metric tons of waste, the majority is, especially of the pieces that you see, are actually microplastics. So you end up having an emerging source of plastic waste, which then becomes more, let's say, available to other ecosystems. Unfortunately, there's about 5 of these oceanic plastic waste collection sites around the world. This poor individual has been trying to escape the plastic waste in the Great Pacific Plastic Ocean for a number of days. But you see, it's just as far as the eye can see. And quite interesting, and I think this really illustrates one of the major problems that is affecting the globe currently, is that if you stopped all input of plastic waste into the ocean right now, in 100 years, 10% would still remain. So in 100 years, you've only got rid of 90%. A lot of this plastic in the ocean defragments and becomes part of the total suspended sediment and the water column, and then becomes more bioavailable. So where does all the plastic waste come? Well, it comes from about 80% of the plastic waste in the ocean is from land sites. So essentially what happens, countries take their plastic waste and export it. And one of the major importers of plastic waste was China. However, China in 2018, based on environmental concerns they enacted a complete legislation which actually overnight stopped the importation of plastic waste. There's been other countries which will go unnamed that have picked up this, this importation, but exactly a lot of the countries that have picked up the slack from China importing plastic waste products, their management is strictly landfilling. So this, this very quickly just identifies some of the plastic resin codes that are used for plastic, and this is the primary means for supporting plastic waste as you relate to potential recycling. And you see that of the two main types that are primarily recycled, which is the polyethylene terephthalate, which is the crinkly water bottles, and the high-density polyethylene, which is the jars that a lot of food come in, they tend to be more recycled, where other plastics, which are very chemically inert and very resistant to any decomposition, are rarely packaged, and they have very long environmental half-lives. For example, the high-density polyethylene, which fortunately tends to be one of the plastics that's usually recycled, its decomposition rate in the environment is about 100 years. The low-density polyethylene is basically non-compostable, so it just will not break down. So essentially what we're saying is that it's just a resin identification. It assists in recycling. Some plastics can be reused, but not all plastics can be recycled. And it's primarily the polyethylene terephthalate and high-density polyethylene that are recycled. And in fact, what adds to the landfilling of plastic waste is that some of these, because the recycling is so difficult, they're not even collected. So this, the primary illustration here is that sort of blue is good, and what we mentioned is that polyethylene and the HDPE are primarily recycled. The yellow is not so good, and the red is bad. So you see that some of the actors which contribute significantly to plastic waste are either rarely recycled or never recycled. So this just provides a bit of information about polyethylene terephthalate, exactly what it's made of. It's very malleable. It can be, it's formed from ethylene glycol and terephthalic acid, but it can be molded into almost any shape. It does have restrictions in terms of the sort of tensile strength and the reusability, but it's very practical in terms of usage. About half the world's synthetic fiber is made from polyethylene terephthalate when it's referred to as polyester. When it's used in food packaging, it's just polyethylene terephthalate, but it's the same thing. So polyethylene terephthalate is almost completely recyclable, except the current recycling rates don't approach, in most cases, any more than 30 or 40%. 50% in Europe, which is quite high, but as a compound, it's totally recyclable. It's not, we'll go over some of the recycling issues, but it's actually relatively easy to recycle. But also, about 40% of the energy that's gone into production of polyethylene terephthalate is recoverable. So that means it's also very attractive for energy recovery. And this is sort of the similar issue with high-density polyethylene. It's a very effective food packaging material because it's very resistant to all different types of food: acidic, basic, fatty, alcohol, etc. And the one good thing about HDPE is that it's one of those packaging materials that it can be continually recycled without seeing loss of functionality. So, and these are some of the older figures from the US that about 31% of HDPE. However, once it gets into the environment, it has a very long decomposition half-life. So why recycle plastics? Well, obviously it conserves natural resources. It reduces landfill use. It helps to reduce environmental contamination. It helps reduce plastics in the environment. There's a financial incentive to actually increasing the recycling because of the the market that's growing for recycled plastics. And more importantly, both food contact material and especially recycled plastics are a source when they get into the environment of micro and nanoplastics. And there's been a great deal of information published about the potential human health effects of micro and nanoplastics, exposure to humans, detection of microplastics in humans. It's been estimated that the actual microplastic content of every human is consuming about the size of a credit card on a weekly basis. And some very recent information has shown that the human brain actually contains about 0.5% plastic by weight, which is sort of unsettling news. So this just basically is a survey that was conducted by Accenture of 6,000 consumers. And unfortunately, one of the main factors that was important to consumers was price, whereas environmental concerns of a packaging material were ranked lower. So we get into the recycling process. Mechanical recycling is the best way. You just collect the process, you wash it, you clean it, you chew it up, and then you reform other types of plastic. So polyethylene terephthalate is one of the major components of mechanical recycling. Chemical recycling is increasing in use, and that's when you break down the plastic into its monomeric components and its constituents. Unfortunately, it's much more expensive. So there's different types of depolymerization and pyrolysis, but it's currently, chemical recycling is about 5 to 6 times more expensive than mechanical recycling. Whoopsie. Well, I guess we can't go back. Anyways, so the general concept for safety assessment of plastics, and some of these are applicable also to recycled plastics, is that all plastics contain residual chemicals used in their manufacture. So these can be a variety of materials used, both the creation of the polymers, plus things that are added to the polymers. So migration occurs under normal conditions of use, so it doesn't have to be extreme conditions of use. This is from an unknown or unnamed federal regulatory agency who looked at the migration of chemicals from packaging materials over the course of about, say, 15 years. So you see that the cutoff point is about half of the chemical migrants are resulting in exposures greater than a nanogram per kilogram body weight per day, which is quite low. But there are some that are up close to a milligram per kilogram body weight per day, and that would be more equivalent to, say, of food additives. So the chemical industry says, chemical migration examples are very simplistic. You have monomers, you're recycling, you have mechanical recycling, and you basically get a safe consumer product. In effect, you get migration of other chemicals that were never meant to be in packaging materials, and these are compounds that were left over from the polymerization process. These are reactor products with the additives that have been added, and also formed during the recycling process, either mechanical or chemical. So this group, which is appropriately named International Pollutant Elimination Network, basically says recycling plastics is simply recycling toxic chemicals. And if you look, this pictorial basically indicates all of the areas where recycled or toxic plastics can be increased exposure, either to humans or the environments from the recycling process. UNEP has identified 10 groups of chemicals associated with plastic as being major concern, and this is from toxicity and migration potentials. And what we now see issues to recycled plastics is that generally, you find that there's a higher volume of chemical migrations coming from the recycled plastics compared to virgin materials. So the other issue is that there's potential consumer misuse of plastics before they're subjected to recycling. So you get introduction of all unknown different types of chemicals. And this may result from inadequate sorting. And then you get degradation products because recycled plastic generally doesn't have the structural integrity as the new products. And this is basically the Safe Food Advocacy Europe group says that recycled plastics specifically from a food contact material exposure chemical, is considered a high-risk issue. So this just basically summarizes some of the chemicals that can be migrating from recycled plastics. One of the more areas, there's chemicals that are directly added to the plastic for a functional purpose, and these are referred to as intentionally added substances. More importantly, you have a wide variety of non-intentionally added substances. So these things are never approved for packaging, but for either reason, they're being picked up from environmental sources, from consumer use, and they are then subject to recycling. So what we see is some of the issues specific to recycled plastics. These are different types of recycled plastics that have been collected, extracted, and then analyzed for a wide variety of chemicals. What you found in this particular study is that the fraction of products with occurrence is greater, and that's illustrated by the white bars, for the recycled plastics compared to the blue bars, which is the virgin material. This is recycled pellets that have been collected from recycling facilities and then subject to just an extraction. The light green is the high-density polyethylene, and the purple is low density. One of the issues that have been identified is that the number of peaks that were prevalent in this recycling plastic material, especially the HDPE, was over 1,000. And unfortunately, these are some of the areas where you break down what are the identification of the different types of peaks. But this comes from approximately only 10% of the chemicals that could be identified from the non-intentionally added substances which were potentially migrating. So issues specific to recycling plastics, you have to consider the fact that these have to be effectively clean, and there could be chemical misuse by consumers prior to the recycling process. So how does the industry currently handle that? They provide a number of chemical surrogates, and these are some of them that are actually added to the plastic material in very extreme conditions in order to ensure that there is limited to no migration of chemicals that may have been misused through the lifespan of this plastic. So we won't go into a lot of detail of this, but this is basically a way that has been used for both the toxicological assessment of unknown migrating substances. The same process is also being used for flavoring ingredients and newly formed packaging materials. It's based on the premise that the toxicity of a chemical can be based on any structural alert the chemical may have. So the issue specific to recycling plastics is that you have to provide a complete description of the recycling process, including the initial collection. And this a lot of times is lost. You have to have effective sorting, which allows food use from food type controls. And you have to show that there's been a complete elimination or neutralization of all chemicals during the recycling process. And that was the chemical surrogate issue I was referring to before. So the conclusions: plastic waste has created a great need for the recycling of plastic materials. A number of countries have produced legislation to phase out of single-use plastics. And the vision is, especially in Europe, by 2030, that all plastics on the market should be recyclable. So we won't go into a lot. We won't go into this. People don't like to say bad things about Coca-Cola, so we'll skip over that. Future actions. Certain countries have developed regulations that approve recycled plastics, including the safety assessment of non-intentionally added substances and intentionally added substances. And these include USA, EU, Canada, and Brazil. Recycled plastics do represent a challenge for identification and control of intentionally added substances and non-intentionally added substances. The good news is that Codex Alimentarius is currently looking at providing a type of guidance for the food safety assessment considerations that would have to be required for recycled plastics. So we're sort of on the last, we're leaving with the good news story for the UAE. In 2024 in Abu Dhabi, there was a restriction placed on the use of plastic bags, which was determined to be over 95% successful. That was rolled out to the rest of the UAE in 2024, which implemented a national ban on plastic bags. In 2026, that's going to be extended to single-use plastics, which makes up a significant contribution of the plastic waste. They have a specific plastic ban resolution and circular economy, which is also promoting other avenues. And the good news is, as of 2027, the UAE has announced that they will be landfill-free through initiatives where they will be generating waste recycling into energy recovery and different plastic recycling type of parameters. They will close all of their landfills to collection of plastic waste. So that's sort of a goal for other countries, especially those dealing with large amounts of plastic waste. And these are sort of just some of my personal opinions. Why isn't plastic recycling more? It's primarily economic barriers. The other thing is that you have industry that is resisting commitments to use recycled plastics and develop materials that can be easily recycled. And then a general concept is that continued production of virgin plastics undermines circular economy. And with that, knowing Amin is going to tell me I've gone way over time, I would end my talk. Thank you.
Thank you so much, Mark, for that comprehensive analysis of this timely issue related to recycled plastics, and especially to these emerging chemical contaminants, the so-called non-intentionally added substances, and even the intentionally added substances that we already know. So just, dear audience, we will not be taking questions immediately after each presentation. Rather, we will reserve all questions for the panel discussion at the end of the session, where we will have all the opportunity to engage with all our speakers collectively. So for our second presentation, please allow me, and together we will welcome Mr. Laurent Auger, Director General of the International Sweeteners Association. Mr. Auger will discuss with us how science and perception are shaping the future of sweeteners in food. Mr. Laurent, the floor is yours.
Hello, good afternoon. Good morning, everyone. Thank you very much to the organizers of this session, but also of the whole event, for the opportunity to present today the view on, I would say, a specific topic, but maybe a good illustration with a deep dive into a certain specific type of ingredients, which are sweeteners, and see how, in the sweetener situation, there's been a clash, I would say, between some of the regulatory science, in a broad sense, with new considerations that has caused consumers questioning. So let me start with a basic outline where I will be discussing the ISA, where I'm talking from, what do we cover as an association, explain very briefly what are sweeteners per se, but then go more in depth into their safety evaluation, but then that's the main part of the presentation, talk about some of the controversies around this type of ingredients. So just as a basic introduction, the ISA is a non-profit organization with scientific objectives, and we represent suppliers of sweeteners as well as end users. You can see here the logo of our members. And by low and no calorie sweeteners here, I refer to those that brings no calories when used in a final product. We are focusing on the sweeteners, as I said, with no and low calorie, and we will not be covering those like polyols, for example, who are still bringing a certain amount of calories, just to be clear. But most of my talk will be able to address sweeteners in general. So quickly, to clarify the situation also and explain basically what they are and what they are not, sweeteners, and in our case, low and no calorie sweeteners, they are basically food ingredients, often authorized as food additives. that brings a sweet taste to a final product when used in formulations. And they bring virtually no or very little or almost none calories into the product. And they are used in place of sugar. That's an important point. It's really as an alternative to the sugar and especially to the calories brought in a formulation by sugar. That's one of the main goals of using such ingredient into a formulation. And as such, only a very tiny amount would be required to bring the sweet taste into a formulation compared to the sugar, if you had to compare the two. And also, just to be clear, when it comes to the regulatory aspect about presence in a final product, I think in almost all jurisdictions, you would have very clearly put on the label the presence of a sweetener, either on the back of pack or in certain countries, even on the front of pack. If I take the example of the EU, you would have the word with sweeteners directly associated, for example, with the main name of the product on the front of pack. So the communication and information towards consumer, I would say, is on a high level if I compare to other types of ingredients. And you know all those categories, they are very common. You would find sweeteners in many of those types of products, including the tabletop, where the sweetener is directly in itself used, either in your coffee or in your tea, for example, or to cook even, why not? But I would just insist here on this slide on one category at the bottom, which is the medicinal product. That's often something overlooked. My talk is really around the applications in food, but that's also something to consider. Sweeteners are often used in lower amounts, clearly, but often used as excipients as well in medicinal products. It's easy to imagine, for example, some bad taste of an active ingredient in a pharmaceutical product would need to be masked, altered, just to make sure you have adherence to treatment by patients. So when we talk about low and no calorie sweeteners, being artificial or being natural, it doesn't change anything here, I would say, when it comes to their safety evaluation or even the benefits. Here you would have a table with the most common ones. Some of the names are very well known. Everybody has seen many headlines around aspartame or sucralose, for example, but some are less known, or even some of the new ones might not be known to you. But the overall list is not so long. It's not exhaustive, but I would say you have maybe 13 to 15 substances in total. What this table shows as well is some of them are very old. Some of them were discovered and authorized a long time ago, showing also there's a long history for certain of them around their safety checks, evaluations, and background information available. We also see their international number, being the INS number for the global standard. This is common for all those type of additives. But I'd like to insist on the differences. Often, consumers would be exposed, and everyone, basically, like us, would be exposed to media headlines mentioning sweeteners are causing this, sweeteners are causing that effect, or whatever, based on certain studies being published and interpreted by media. Unfortunately, if you go deeper and look at the article, often you realize the underlying study would be just based on one substance tested, or two maximum. So just to show that all those substances are also very different, and it's a bit a reduction to often talk about the whole category. And for example, you see the sweetness equivalent. If you just compare their potency to bring a sweet taste, some of them, when measured compared to sucrose, so sugar, would have thousands, 10,000s of times more potency to bring the sweet taste, meaning you also need a tiny fraction in milligrams into a final formulation to have the same level of sweetness. So the potency of sweet taste would be very different. Same goes with general properties. They don't react the same way. They don't have the same fate. Some break down very easily in the metabolism. Aspartame just breaks down in your stomach, basically, in free natural constituents. Others would go and be excreted by the body completely, almost untouched, like sucralose, for example. So that's fundamental differences, fundamental properties which make them differ from one to another. Those properties also affect how they are used. You don't use the same product if you do a solid formulation compared to a beverage formulation. The consideration would be very different. And maybe one last point as well is around the taste in itself. There are major differences, and we all have, I'm sure, some of our own preferences. Some might leave an aftertaste, for example. There's some I personally would dislike and prefer another. So that makes those properties that makes the substances very different from one to another. And I think we should just sometimes avoid falling into the trap of calling the whole categories. Just quickly, and before going into safety, when we talk about their contribution of sweeteners when it comes to public health, I just mentioned the first two, which are very easy because we're already covered. The reduction of energy intake is a given. That's one of the main reasons of using it in replacement to sugar. You don't bring in the calories. So also it means if you want to support a specific strategy to lose weight or maintain a weight loss, that can be an appropriate tool. But they also have other benefits. Here, for example, when it comes to blood sugar levels, and for example, people suffering from diabetes, they can be a tool to be used to still enjoy a sweet taste despite having a condition, for example, like diabetes, because they do not affect your blood glucose or your insulin level. So it's much easier for those patients to monitor their intake of food. How much does it cost to bring the sweetness in a product in a formulation? How much does it cost to bring the sweetness in a product in a formulation through sugar if you use a sweetener instead? What does it mean in terms of land use, water use, energy use, all those parameters of life cycle assessment? There is also things to look at on the sweetener side. So on the safety, because consumers, I think that's maybe the first question we keep getting from general population and consumers around safety of sweeteners. Just a reminder here, I think it's well known by many colleagues in the room, being food additives, they are evaluated. For example, at the global level, there are several entities, regional and global level, taking care of their evaluation. If I look at the available set of data, especially for the main sweeteners, thinking aspartame, for example, the history and the number of studies is rather impressive. The size of the dossier is very consequent. I would even say today, the scrutiny level that sweeteners have been gone through to date, I'm talking the existing ones, might be one of the highest for a food ingredient, clearly for a food ingredient. So at the global level, it's the competence of the joint World Health Organization and FAO expert committees, the JECFA, to oversee their evaluation, but we also have dedicated programs like in Europe, but also in other jurisdictions like in the US or Australia, New Zealand, or even Canada. If we look at what should contain a basic dossier and a basic safety check package, that would be the general categories. I won't go into all the details, but when people and consumers get questions around, does it give cancer? Is there an issue on this aspect about absorption, etc., you would have this kind of information already part of your basic information package that needs to be submitted, being at the global level or being in the regional level. I would insist on this point here on epidemiology data. We often get a comment saying it's only industry data that is being looked at and not that data from academia or what exists out there in the literature and which was not proactively pushed to the regulators. That's not entirely true. If I take the example of the European Union, for example, alongside the substantial dossier a company would need to provide EFSA, or when EFSA is calling for a review of an existing substance, there are literature searches, deep, extensive literature searches, which are also conducted, making sure to try to capture latest information. In the case of sweeteners, that also includes all the volumes coming more from the nutrition science side. So, on the basis of those information, authorities would also have a look at considering specific cases. I'm thinking here, what about kids? What about elderly people? What about pregnant women? And for example, and those would be part of the evaluation effort. And with this, also in conjunction with work on exposure and predicted exposure or measured exposure, some values like an acceptable daily intake would be set. I won't go into detail on how it is formed, but basically starting from lab studies showing a no effect, making sure safety factors are embedded in to account for all various situations, and ending up with a number. And today, such number is set for all the sweeteners authorized, and if we look at the global level, all the studies measuring actual intake of sweeteners would be below those ADI. I think it's very reassuring to see that studies confirms that we are still below those levels. It doesn't mean some studies doesn't have a couple of outliers. You can still find some, not artifact, but at least a measure point showing one person one day went over the ADI. That's okay. And that's part of our role also to explain to consumers and general public, it's okay in a sense. It doesn't mean a safety issue. It's an indication, it's a threshold value. It's not a safety value and a safety threshold per se. You can go over the ADI, but the ADI has been built from the ground based on data to also account for your full entire life exposure at such high level. So if you're ending up above the ADI, if I take the example of a diet soda with 200 milligram of aspartame, the ADI is 40 milligram per day per kilo. If you take a 65 kilogram, not me, but body, a young woman, for example, it means more than 14 cans per day to reach the ADI. But then that would assume if you really want to have an issue, you would need to consume this number of cans every day for your entire lifetime. And still, you would be not at the safety concern level, but that would be an indication. So I think you have other problems if you're already at that level of consumption per day. So just to show that this kind of concepts are embedded straight into the initial evaluation of those substances. As an example of how the science is trying to also considered and being updated. Yeah. This is an example of the EU re-evaluation program. In 2009, it was decided to go into a re-evaluation of all additives in Europe. Sweeteners were given preference and priority, I would say. And today, we are still missing only 3 of the low and no-calorie sweeteners. And you have the timetable on the screen here. Sucralose, the salt of aspartame, acesulfame, and cyclamates should be coming later this year and beginning next year. But so far, all of the others were confirmed in their safety, considering up-to-date information. And even the last 3, saccharin, neotame, and acesulfame K, from last year and this year, we even had, I would even say, the positive surprise to see EFSA, the European Food Safety Agency, increase the ADI level, just because more information is available. The initial dossier were made in the 90s. Now there's much more background, there's much more information showing it was maybe a bit too conservative back in the days. So here at least, it was confirmed with new levels. But when it comes to controversies, and I'll continue on this. One of the main ones that triggered lots of consumer, I would say, confusion came from the YARC classification of aspartame. I think you all remember in 2023 this work. I know some colleagues will maybe go deeper in the difference between hazard and risk. Despite having colleagues from JECFA producing a risk assessment at the same time, and both conclusions of JECFA and IARC were released, I think, on the same day, in even a joint press conference. Unfortunately, the only thing that was, I would say, higher on the media news and headlines was the YARC classification. So YARC looked only at the hazard, and JECFA took everything into consideration, the hazard properties plus the actual exposure, leading to a proper, I would say, risk assessment. That made the YARC classification in the same category as 2B, based on limited evidence here, as a possibly carcinogenic to humans. And here it puts it in the same category as pickled vegetables, for example. And we know WHO IARC, is not calling for people to stop consuming kimchi, for example. Far from it. So just to put things into perspective, but that's one controversy we still have to date to answer to, I would say, despite the clean risk assessment, I would say, produced in parallel. Another type of controversy comes from new studies, especially on those two topics. One is on gut microbiome health, one is on exposure to multiple sweeteners, all those cocktail effect headlines. On gut microbiome, that's true. Initial studies from 2014-15 were very interesting in pointing the finger at a new area being developed of research. We needed to know much more about what happens. I think it's the case in many other areas, and not only for sweeteners, where gut microbiome is being explored. Although now, 2025, and in the last couple of years, we start having more robust randomized controlled trials result. And even the last one from, I think, 3 weeks ago, this Pang et al. Paper, showed actual proper results confirming there's actually no real risk coming from sweeteners consumption on microbiome health. Same with this cocktail aspect, which is often pushed by certain players and also some media. We still see today no valid sign at the scientific level that there would be, for example, any synergy or any specific effect linked to the combination. Those substances sometimes are just way too different. They don't behave the same, as I explained. So you may have seen those kind of headlines. I'm just here using a couple of examples. And one very recent one, even back in September, was around this cognitive decline. 1 can per day of diet, whatever, would lead to 1.6 years of cognitive decline. It's based on a very recent study from September, made in Brazil, based on a cohort following people. But that's also this type of papers which are often fueling consumers' confusion. We see questions coming mainly from observational studies. So there's a mismatch, in a way, between all the randomized controlled trials which are confirming certain aspects, weight loss, no impact on glucose level, etc., with some other results coming just from statistical associations derived from bigger observational studies. So we get it. It's very difficult to get randomized controlled trial in place. It takes a long time, costs a lot. But we know in the pyramid of evaluation of studies and evidence, ranking of evidence, they are also clearly higher. So just to highlight some of those studies, and here again the example of the Nutrinet cohort in France, are just showing a statistical association and not at all a direct cause-effect relationship. One key point as well, in 23, the WHO recommended to not use sweeteners for weight loss and to reduce the risk of non-communicable disease. I have to highlight here, it is a conditional recommendation. It is defined by WHO itself as recommendations which are limited and are of low certainty. And they need, according to their own text, substantial discussions before being translated into policy actions. And we've seen in that guideline development working group that further weight was given to observational study result rather than randomized control trial. That's a bit unfortunate. But they were also calling for more of those research control trials. And that's something we start having. I mentioned this Peng et al. Paper, but 3 weeks ago when it was published, it was a long term, I think that the first ever so long, I would say 12 months, which is very extensive for a randomized control trial, around weight loss funded by the EU and showing actual positive result in terms of weight loss maintenance over the 12 months period. So the data called for by WHO is available. And also in a similar way, when we look at the recent advice from the UK Scientific Advisory Committee on Nutrition, when looking at the underlying science on sweeteners, they would conclude differently from the WHO today based on the information now newly available. So that's interesting to see things are evolving fast. And maybe just one point as well to conclude on controversies. We often get also the question around sweet taste exposure. There's this common, I would say, almost a common sense idea people may have. If you're exposed to a sweet product, you will want more. You will crave more for other additional sweet products. Problem is, it's a bit difficult to measure, and there's actually a lack of data on this, a lack of scientific studies showing such effect. So it is an intuitive idea, but to date, it's really difficult to get this from an actual measurement. So what we see from systematic reviews, but also emerging trials, and for those of you who were maybe in the International Conference on Nutrition in Paris in August, we've seen presentation of about to be published result from Wageningen University in the Netherlands with a very interesting long-term randomized control trial about sweet taste exposure and showing basically people doesn't have their sweet taste preference affected even after a very high exposure to sweet or a very low exposure, depending on the groups. So something to watch for. But it still needs work to clarify this relationship because despite being intuitive, the idea, it doesn't mean it's true. Problem is those controversies are making their way into policies. I'm here just using a couple of examples based on things we've seen. You've seen this kind of labeling policies being those warning signs you have in Latin America, in Colombia and Mexico would have those warning signs with sweeteners not recommended for children. Today, I would fundamentally disagree. There's no real high quality data showing there's a concern. But that's a policy that has been selected. Same goes with the voluntary scheme of Nutri-Score used by a couple of countries in Europe, 7 of them only. In its latest algorithm update in 23, there was some negative points given if a sweetener is present into a product. Okay, that's their decision. For us, it is not backed by science, and actually it would even penalize products which are trying to bring a positive element to the general goal of public health of reducing obesity. And same goes with, and that's the French Assembly in picture, same goes with some taxation policies. When done right, and I would take the example of the taxation policy in the UK, affecting sugar in sugary drinks, you would take off the market vast amounts of sugar with a positive result on your objective of public health. Unfortunately, the French General Assembly a couple of years ago decided to impose a similar tax, different calculations, but in the same scope, you have also the tax applicable to products with sweeteners. So clearly killing the incentives for companies to reformulate, but also creating a difficult situation with products even taxed. So people, for example, as I mentioned with diabetes, who want to access certain products would also have to pay more. So this it undermines also the whole objective of public health, if your public health objective is to fight obesity, but also something that is of concern to me, it might even create issues and sow doubts into the proper work of your scientific evaluation authorities. when they've worked on dossier, checked all the science, coming with those kind of policy, casting a negative light on a substance, and only this substance, I have to say, not other types of ingredients, just this one, that's a bit unfortunate. And with this, I would just encourage all of us to work together to move the sweetener science forward by having this kind of exchange of presentation and allowing we have the proper forum. And with that, I would thank you for your attention and invite you to check our material on our website, sweeteners.org. Thank you very much.
Thank you, Mr. Roger. Your insights highlight one more how science and communication are central to consumer confidence and trust. Let's now broaden our view to the methodologies underpinning chemical risk decision-making. And here we are fortunate and honored to have Professor Bensu Karahalil, Vice President of Turkish Society of Toxicology and Head of Toxicology Department at the Faculty of Pharmacy, Gazi University in Turkey. Professor Bensu, the floor is yours.
Thank you very much. I would like to thank the organizing committee to invite me to share my information or experience. I think the key issue to know the quality of data. First, I'm gonna say a couple of sentences to introduction sentence. We know that various chemicals in present foods in a variety of reasons, such as naturally occurring and also during the process, there are some chemicals takes place. And food safety decisions are based on a large amount of data. Regulatory agencies must determine possible risks to actual exposure levels. And the best way for global food safety is risk assessment. Everybody knows that. The best approach for evaluating all available data, reducing bias, and supporting repeatable regulatory outcomes is the weight of evidence. Risk assessment is a priority issue for all international and national health authorities because international collaboration in food safety is essential. There is a consensus, should be consensus among the health and food authorities and scientific organizations. These authorities provide credible information to us. The risk assessment is essential, and also risk analysis consists of 3 steps. First steps of Lauren already mentioned that, science-based risk assessment. The second one is policy-based risk management and risk communication, which covers all of the steps. But before the risk assessment, I would like to show the difference between what is hazard and what is risk. Let me give an example to explain this one. A hazard, something has the potential to harm you. For example, a shark in the sea is a hazard. Swimming with a shark is a risk. It means that risk is the probability that a hazard will cause harm. We are talking about the food and food safety. It means that hazard in food, a factor that leads to adverse effects, and also risk, a function of the probability of the adverse effect and the magnitude that from hazard or hazards in food. How are hazards and risk regulated? The risk assessment. The risk assessment consists of 4 steps. First step is hazard identification. We obtained the data from toxicity tests and also published papers, and we use this data. Is substance can be harmful? If it is harmful, what does it cause? And we can answer this question. The second one, hazard characterization. what kind of effect cause hazard? We estimate the severity and duration of the hazard. And the third exposure assessment, how amount exposure, how long exposure, how often exposure, who can be harmed, we can get results, response these questions. And the last one is risk characterization. We get informations all about steps and get together, evaluate, and report to the risk managers, those helping them make decision. For risk assessment, we need a database, science-based data, and also data is obtained from toxicity tests. There are some OECD guidelines. When you look at the OECD guidelines, which kind of toxicity test we will conduct, you can see the step-by-step how to conduct the experiments. And we apply toxicity tests to each food products requiring safety before authorization. After toxicity tests, we obtain some numerical values, such as NOAEL and such as other. Lauren already talked about the other. Just I want to quick look. We get the NOAEL. NOAEL is the highest dose that shows no observed effect levels in experimental animals. It's used to most sensitive critical health outcomes in the most sensitive animal species. And this value is used for animals and obtained from the animals. But we need a dose in human use. That's the reason we are using the safety factors. We generally use safety factors 100. 10 of the 100 is used for conversion from animal data to the human data, because we know that there are some toxicokinetic and toxicodynamic differences between animal and human. And the other 10 in safety factor is used for conversion human to human. We know that we are different from toxicodynamic and again toxicokinetic properties change from human to human. This is very important to talk about the data quality. This is an evidence pyramid, data pyramid, hierarchic, scientific evidence. There are many names. When you look at this pyramid, you will, when you go to the upper levels, you will see the quality of evidence is increased and reduce bias, and also trust of causality is increased. So I mean that the upper levels are much more weight, much more valuable compared to the baseline, to the top of the layers. Evidence-based research analyzes multiple experimental and observational studies. Of course, experimental studies much more stronger than observational studies. Why? Because experimental studies, in this type of studies, researchers control an intervention, randomize, allocate groups to the different treatment groups, preventing bias, and also allows causality. But this kind of experiments, sometimes expensive, take a long time to conduct, and maybe sometimes it's not feasible. Generally, for example, randomized control studies is experimental studies. Very few randomized control studies conducted on diet, because participants are put on diets that are short-term and do not reflect the real life. On the other side, observational studies, there is no intervention. They just identify existing group that already have a dietary habits, just only response. And also this kind of studies is much more sensitive, susceptible to scientific bias. We know that scientific bias is we cannot control the factors, and these factors are very important because these factors can change the results, study results. And also causation and correlation. What is the difference of all these two terms? Causation, one thing causes another thing happen. Correlation, when two or more things appears to be related. Let me give example. Ice cream sales increase, shark attack increase. Does this mean eating ice cream increases risk of shark attacks? No. And also, eat ice cream, shark attacks, there may be a correlation between eating ice cream and shark attacks, but one does not cause the other. Correlation does not always mean causation. We have to be careful to make a decision. And to understand, it's essential to understand all this data, scientific data correctly, when experimental evidence, which is the gold standard, may be lacking or limited, regulators may turn to the observational evidence. For non-scientists to interpret correctly for evidence, it's essential to understand methodologies, results, and limitations. And what about the weight of evidence? When you look at this data pyramid, I can put on the top weight of evidence. Why? Because weight of evidence, different types of data alone may not be sufficient to comprehensively assess the effects of substance. Data integration approach, weight of evidence, provides more robust evaluation. As a systematic method, evaluate and integrate all available scientific data, including inconsistent and incomplete data, and also to make regularly decisions. Multiple data sources are gathered and also evaluated together. For example, if you have got multiple randomized control studies and meta-analyses, it provides us strong evidence. I can say that high weight of evidence. If there is several cohort studies provide stronger evidence, I can say that moderate weight of evidence. If you have a study, just a single case report or expert opinion, these are not provide strong evidence. I can say that low weight of evidence. The weight of evidence approach does not treat all types of evidence equally. More reliable, more relevance, more consistent, more accurate evidence is given greater in the decision-making process. The weighting systems helps prioritize data while considering the limits of weaker evidence. What about the principles of weight of evidence? Relevance is very important. The evidence relevant to the hypothesis of study. For example, doses relevant to the real-world exposure levels. The second one, quality and reliability, is important. Each evidence should be evaluated in terms of scientific quality. What is the design? The sample size is large or small, and statistical power is high or not. High-quality studies carry more weight. And the third one is consistency and coherence. Findings from different studies should be consistent. Consistency increases the reliability. And also strength and magnitude of evidence. The strength of observed and associated should be evaluated. And also the first one, biological plausibility. Evidence must be consistent with known mode of action, science principles. And transparency and documentation, also important. The entire weight of evidence must be clearly documented, and decision must be traced and reproducible. The integration evidence Any multi-evidence should be combined according to its relevance and quality, uncertainty and limitations. Is any uncertain or data gaps? What effect happened the reliability of the results? And also objectivity, it's important. We have to avoid the bias. The assessment should be objective and fair. Avoid cherry picking, I mean no selective data use. The last one is weighting and integration framework. Evidence is weighting, such as quality, quantitative, according to the criteria. Which kind of criteria? Reliability, relevance, consistency. It should be produce a transparent, logical, and justified conclusion. And also the other important thing, we have to actually for non-scientists to know how to read and evaluate the scientific paper. When you look at the paper, you will see the basic overview on the abstracts and how to select which kind of study type. And we have to know it's a scientifically powerful and is there a conflict of interest? We have to know all of them. We can evaluate. Policymakers, how they understand the severity of different risks, what level of risk merit regulation, different authorities apply different risk thresholds. Risk thresholds subjective, but still benefit because benefits to compare the certain common risk. The second one, risk matrix is created to identify which risk is very harmful, which risk is less harmful. The third one, risk prioritization among different risks according to potential impact and probability of occurrence. For effective policy, policymakers need to know cause of particular outcome, a conclusion that cannot be drawn from only observational evidence. Again, ice cream is here. And let me give the example. Policymakers considering how to decrease the risk of injury from shark attacks might be presented with the evidence that eating ice cream is associated with increased shark attacks. Ice cream does not cause shark attacks. We know that swimming in shark invested waters causes shark attacks. This situation may occur on hot days when people eat ice cream. The important thing that without addressing the cause, it would not be right to make policy that reduce ice cream consumption. What remains in our minds? The best way for food and health safety is risk assessment approach. The use of evidence-based data is essential for risk assessment. The weight of evidence approach enables robust regulations since it evaluates and integrates all available scientific data, leading to more reliable, consistent, and accurate outcomes. Understanding and interpreting scientific studies are also critical for risk assessment since risk assessors provide information to the risk managers to make legislation. Thank you very much for listening.
Thank you, Professor Bensu, for clarifying how diverse data streams inform rigorous decisions. And now, from methods and methodologies to real-world application, let's hear how risk assessment is practiced into the regulatory context. And we are honored to invite our final speaker, Dr. Intisar Al-Gharibi, Director of Risk Assessment and Food Crisis Management in the Food Safety and Quality Center of the Ministry of Agriculture, Fisheries and Water Resources in the Sultanate of Oman, and Chairperson of the FAO-WHO Codex Coordinating Committee of the Near East. Dr. Intisar will share a compelling example of cadmium risk assessment in abalone, illustrating how science directly informs national regulations. Dr. Intisar, the floor is yours.
السلام عليكم ورحمة الله وبركاته. أسعد الله مساءكم بكل خير. Dear colleague and distinguished, I'm going to change the language to the Arabic. طبعا سعيدة جدا أن أنا أكون اليوم موجودة بينكم، وأشكر الجهة المنظمة على دعوتنا لحضور هذا المؤتمر. طبعا محاضرتي اليوم راح تتكلم عن تجربتنا في عملية تقييم المخاطر في الأبلوني. بتكون المحاور اللي راح أتحدث عنها طبعاً مقدمة بسيطة عن الأبلوني، إيش هي المشكلة، إيش الأهداف اللي تمت عليها هذه الدراسة، وطريقة التحليل والنتائج والخلاصة أو التوصيات. طبعاً نتكلم عن الأبلوني، هو عبارة عن عضو بحري رخوي بصدفة واحدة. وطبعاً هذا الأبلوني بالنسبة لنا هو عالي القيمة الغذائية وأيضاً اقتصادياً. مع ذلك وجوده يعني من فئات المحار، يعني ممكن يكون الكادميوم موجود طبيعي بسبب البيئة اللي حواليته، ومنها المعادن اللي تكون موجودة هي مثل الكادميوم. أيضاً التحديات اللي واجهناها أنها لا توجد حدود أو ليميتد في الكودكس إليمنتري، عشان ممكن نقيم أنه هذا فعلاً النسبة الموجودة فيها هي تعتبر خطر أو لا. طبعاً هناك الفجوات التنظيمية، أنه يجب أن يقيم سلامة هذا المنتج. طبعاً هناك سؤال تساءلناه قبل البدء في عملية التحليل أو عملية التقييم: هل هذه المستويات اللي تم الكشف عنها هي عالية الخطورة أو مضرة بالصحة للمستهلكين؟ كان هذا بداية عملية التحليل. طبعاً إيش كان الهدف من تقييم هذه الدراسة؟ اللي هو لتحديد ما كانت هذه المستويات اللي تم العثور عليها من خلال النتائج في عينات الأبلوني، هل هي تشكل خطر أم لا على المستهلكين، وأيضاً لمعرفة أنه أو لوضع حدود تتناسب لاستهلاك هذه الأبلوني. طبعاً في إطار اتخاذ القرارات في مثل هذه الدراسات، أولاً عند الكشف مثل هذه المستويات اللي هي المرتفعة في المعادن الثقيلة، ولا توجد هناك حدود مرجعية ممكن الرجوع إليها، وبخاصة أنه في الكودكس ما في توجد حدود للكادميوم في الأبلوني. فمباشرة تم عمل تقييم اللي هو إجرائي سريع في تحليل المخاطر. كانت من هذه الإجراءات اللي تم العمل عليها أيضاً مراجعة أنماط الاستهلاك، أيضاً تقدير المستويات التعرض الاستهلاك سواء اليومي أو الكمية التي تستهلك، أيضاً مقارنة هذا التعرض اللي يستهلك بالمستويات الأخرى اللي هي موجودة على المستويات الدولية بنسبة استهلاكهم اليومي أو الشهري أو غيرها، ومن بعدها يتم اتخاذ القرارات في تقييم هذا المنتج. طبعاً النتائج اللي رصدناها كانت هناك مجموعة من النتائج، فأخذنا متوسط وجود الكاديوم في هذه العينات اللي هي بلغت 1.5 ملغرام بير كيلوغرام، وأيضاً هناك كانت نتائج للأعلاف التي تستخدم، لأن هذا الأبلوني كانت عبارة اللي هو المستزرع، الأبلوني المستزرع. فأيضاً وضعنا احتمال بسبب وجود يعني بسبب الأعلاف التي تغذى بها هذه الأبلوني وجود الكاديوم أيضاً موجود فيها، وكانت نسبته اللي هي 0.8. طبعاً أيضاً هناك تساؤل آخر: هل هذه المستويات وجودها بسبب أنها هي كانت من مصادر خارجية أو إنها كانت موجودة طبيعية في الأبلوني؟ طبعاً احتمال أن يكون يعني المصادر يعني مختلفة لأنها دخل فيها الأبلوني من خلال النتائج اللي كانت عندنا. بعدها طبعاً من بداية ما عرفنا هذه النتائج والأسئلة التي تم طرحها خلال عملية التحليل، أيضاً قمنا بعمل مقارنة دولية مع التشريعات الأخرى الدولية. طبعاً وجدنا أنه هناك مثلاً كان يستخدم في الصين بنسبة أعتقد 0.4 إلى 1.5 من من جزء في المليون. وأيضا يستخدم في كوريا بنسبة كان المتوسط اللي هو 0.6 كحدود قصوى فيها. طبعا أظهرت هذه النتائج ما زال المستوى اللي تحصلنا عليه ما بين هذه البيانات اللي هي موجودة دوليا. مع ذلك إن المستويات الحالية يعني التي ظهرت ما زالت في الحدود ما بين من 1 إلى 2 جزء في المليون. وكنا نعملها بهدف أن نتحقق من الاتساق بأن النتائج معنا مع المقارنات الدولية. طبعا التعرض للمخاطر الصحية رغم أنه وجود إمكانية لتقييم هذه المخاطر الصحية بسبب نقص في البيانات الاستهلاك اللي هي كانت موجودة عندنا، كانت لا توجد أصلا كميات أو نسب الاستهلاك للأبالوني في عمان أو ما للتقدير يعني بالكميات هذه. حتى عند أعلى المستويات اللي هي المستوى الكادميوم اللي هي كانت 1.5 من نسبة الاستهلاك. طبعا حددنا أنها أخذنا اللي هي نسبة الاستهلاك من دولة الصين، وهي كانت من الدول اللي معروفة باستخدام الأبالوني، فكانت نسبة الاستهلاك بالإجراء باليوم أو بالشهر اللي هي كانت 0.3 وكان في حدود المقبول من وزن الجسم، وهي بناء على اللي أيضا حددتها اللي هي الجفكة فإنها هي المستويات المقبولة. من بعدها طبعا بينا هذا الشيء أنه هناك ما توجد أي مخاوف صحية من وجود هذه النسبة الاستهلاك اليومي. طبعا التوصيات اللي أخذتها إدارة المخاطر بناء على النتائج اللي بعد مع عملية التحليل أنه يجب أن نتأكد من إجراء تحليليات أخرى أو معرفة إيش الإجراءات التي تمت داخل المختبرات، إيش أنواع التحليل اللي تم فيها، هل تم تحليل أبالوني بعد إزالة جميع الأحشاء اللي موجودة عليها، هل هي كانت مجففة أو غيرها. أيضاً نحتاج إلى إنشاء بيانات محلية اللي هي معرفة نسبة الاستهلاك، كم النسبة كم الحدود التي تستهلك خلال اليوم، وأيضاً الاستمرار في الرصد يعني الدوري للكادميوم في العينات الأخرى أو في المنتجات البحرية الأخرى، وأيضاً النظر في معرفة الحد الأقصى اللي هو لنسبة الكادميوم في الرخويات مفصولة عن الرخويات ذات الصدفة الواحدة. طبعاً استخلصنا هنا إنه نسبة استهلاك الكادميوم اللي هي كان تم اكتشافها إن هي ما زالت في ضمن النطاق الطبيعي، أي ضمن الحدود التي تم مقارنتها أيضاً مع الدول الأخرى، وإن هناك لا يوجد أي من دلائل تشير إلى أن النتائج يعني غير عن خارج المصادر أو مصادر بشرية أنه تمت إضافتها كأنها هي توجد بصورة طبيعية، وأنها لا تشكل خطر على الصحة في ظل معلومات الاستهلاك اللي تم إيجادها. ومع ذلك ما زلنا بحاجة إلى المزيد من البيانات اللي هي عملية الاستمرار في الرصد، عملية المتابعة، أيضاً وضع تصنيفات للعينات، وأيضاً نعمل عملية تنظيمية لمعرفة الحدود أو النسب المحددة في الرخويات، وأيضاً من خلال معالجة الجوانب اللي هي تعزيز الحماية صحة المستهلك ورفع اللي هي المستوى، لأن هذه الأشياء تساعدنا مستقبلياً في عمليات التقييم لمثل هذه المنتجات التي لا توجد لها حدود في الكودكس. وبهذا شكراً جزيلاً لكم.
Thank you, Dr. Intisar. This case study shows how targeted deterministic risk assessments can drive safer outcomes in national food safety discussions. And with that, at this point, I have the honor of inviting my co-moderator for our next panel discussion, Dr. Betul Vesketcher, Vice Chairperson of the Codex Alimentarius Commission, to join me on the main stage. I'd also like to welcome back all our speakers as we transition into our panel discussion and open the floor for questions. Kindly, our esteemed speaker, if you could join us back to the main stage. Dr. Betul, the floor is yours.
And thank you very much, Dr. Amin. And it's a great privilege and honor to join this panel and to be part of such a rich and timely discussion on emerging chemical hazards and risks. I would like to begin by expressing my sincere thanks to the organizers, G-Force, and the Dubai International Food Safety Conference, and for bringing us together and to putting together a program that reflects both the urgency and the complexity of food chemical safety issues. And today's representations have highlighted how science, policy, innovation, and public perception intercept in shaping our risk management frameworks. As we move into these panel discussions, I look forward to exploring these themes further with our distinguished speakers and, of course, with all of you audience, and to reflecting on how we can continue advancing collaborative and science-based approach in this field. I would like to thanks again to our speakers for their fruitful presentation, and let me take my seats and to start our panels. Thank you. Thank you very much again. And I hope I will start with a question to all speakers to warming a little bit the podiums. And dear speakers, and to all of you, my question, and how can regulation best anticipate and adapt to emerging chemical hazards in food systems? What proactive actions are needed in risk assessments or even at the risk management level? Yes. Which one do you want to take first? Mark, maybe.
Thank you for that insightful question. I think primarily it's being able to rapidly address unknowns when the situation presents itself from a perspective of unknowns that have never been previously detected in a particular food. So in certain cases, these unknowns may have limited hazard or toxicological data to support any type of risk assessment. So you would have to look at the development and use of alternative strategies. These new types of methodologies, such as comparison based on chemical structure, et cetera, or screening techniques based on computerized programs which have a large database of chemical structures and the known toxicity. I think it's not imperative, but it allows you at least to attempt to develop a coordinated response, which in certain cases would be based on some certainty that the lack of data isn't preventing you from making a reasonable decision.
Thank you, Mark. So you touched to a very important issue related to the lack of data in this regard. So again, I would like to ask our speakers if they want to add anything in this regard before we move to. Please, Professor.
Maybe I can say a couple of things. These are the four new chemicals. So the new chemicals and also new chemicals in the market and newly developed chemicals, it's gonna be a problem. Why it's gonna be a problem? Because we have not enough toxicity data. We have maybe limited information about mechanistic studies. So we have just only in vitro studies and read across similar studies. So we have, it's difficult to determine this kind of data, which one is the available. It's very difficult, this one. And also some studies results is conflicted. For example, there is one laboratory says that, yeah, it has an endocrine activity, endocrine disruptor activity. The other one, it's not. Because this conflict of results, we cannot decide which one is the correct, which one is not correct. So actually regulators decide to judge which one is the valuable for us, and this also creates political sensitivities, because the industry says it's a safe, maybe academics say it's not safe. So it's very important to decide the weight of evidence and weight of studies.
Thank you, Professor Bensou. So perhaps we move to other questions in order to benefit from the presence of all our speakers. So I will go back to Mark, since Mark opened the ball. So Mark, your presentation highlighted, and you already mentioned this, but if you could give us more insights about this issue, especially related to the case or the problem of non-intentionally added substances and contaminants entering the recycling stream. So given all these challenges that you highlighted in your presentation, what do you see as the most critical data gap and or research priorities that need to be addressed to enable a proper assessment of these materials?
Thank you, Amin. It's a very good question. In fact, it's even related to the initial question about how to address, in certain cases, lack of data that is preventing you from making a reasonable scientific decision. In this particular case, what you're faced with is essentially any migrating chemical from packaging materials into food will either be classified as an IAS, and that's substances that are intentionally added. So these have been added to impart a function to the packaging material itself, either in terms of color, use, properties, etc. And in most cases, those go by a standard risk assessment process. You would have to determine the likelihood of migration. You know the chemical structure. You generally do have hazard data to draw an assessment of whether or not that chemical is safe or set specific migration limits, as certain jurisdictions would do. What I see as a larger data gap is the non-intentionally added substances, and I apologize, but I glossed over the example where you had both the high-density polyethylene and the low-density polyethylene, which had been extracted, and all you see by GC mass spec is peaks, and these peaks would represent a potential chemical migrant, and in certain cases, in this particular example, there was over 1,000 peaks that were identified as potential migrants. However, when they compared the spectral graphs to known reference material, they could only identify about 10% of those peaks. So theoretically, to me, a large source of uncertainty is how you accurately identify all of these potential non-intentionally added substances. But the other question is, do you actually need to identify these non-intentionally added substances? Because through the process, and once again, unfortunately, I glossed over it, was this threshold of toxicity idea that has been developed based on identification of structural alerts for chemicals that would potentially have potential toxicity. And this results from extensive evaluation of chemicals that have been evaluated over the years as vet drugs and pesticides and contaminants and food additives. And they all fit into a specific chemical class based on structure with a potential to have a certain type of toxicity. So what this threshold of toxicology has developed, and it's currently being used for the safety assessment of flavoring ingredients. What it shows is that without the identification of a particular chemical, you could define an exposure limit where you have reasonable assurance that that chemical is not going to cause toxicological effects. But whereas that addresses the actual data gap of not being able to fully identify all non-intentionally added substances, it does promote the continued updating and supporting of the threshold of toxicology system, which is in use in a number of countries and a number of organizations. Both EFSA, FDA, JECFA, and Health Canada have adopted this as an approach for both flavoring substances and for food contact notification chemicals. But it always requires an updating of the database to ensure that you've captured all chemical classes and you have reasonable certainty that any not intentionally added substance that could potentially migrate, which you have an unknown structure for, would not represent a health risk.
Thank you for this enlightening answer, and absolutely this TTC approach could bring a solution to tackle this issue of non-intentionally added substances. Thank you. And now I go back to my co-moderator, Dr. Betul.
Thank you very much, Mark. Every kind of assessment is important and started with the quality of the data, and also you mentioned about the quality of toxicological studies. Thank you very much. And I would like to turn to Dr. Intizar, and it's a very fruitful presentation because it is coming from the field. It's the case study. I think it's the very good, the starting point. And your case study clearly demonstrated how the absence of codex limits requires a rigorous database, science-based, nationally, sometimes internationally risk assessment process based on your experience with cadmium in abalone, monovalve mollusks, which is monovalve mollusk, abalone, and what would you consider the most critical data needs, and whether consumption or toxicological points to support a reliable and defensible chemical risk assessment and international reference limits. There is no international limits. Thank you.
Thank you, Albetul, for this good question. From my views, if there is no any codex limited, we need first of all data analysis is very important to collect that data, and we need to know these data done in the labs and the method they use it, how the way they did it in the lab. And the second, and is very, very important, it is consumption data. When we have lack of consumption data, if also there is no any available data in the consumption food, it's very difficult to do our risk, risk assessment, because in the consumption data we can know the quantity, frequency, and sizes of the food they use it, and we can use it in our risk. And the third one is the toxicological data, because it's come from define our health base, and we should get these data from the trust bodies like JEFCA and other body organization for that. And all these three, they are very important for us to do the risk assessment. Thank you.
Thank you very much, Dr. Intizar. And then I'd like to turn to floor the Dr. Professor Bensu. And you have also emphasized the importance of integrating toxicological and epidemiological data through a weight of evidence approach. And in your view, what are the biggest challenges regulators face when applying this approach in real-world decision-making, and especially when dealing with emerging chemicals where data may be incomplete or there's some inconsistency between data? And what's the situation? Thank you.
Thank you, Betul. Actually, there is some challenge. For example, study quality is very different from laboratory to laboratory, and academic studies And also, for example, industry studies standards, they are using TGLP rules, but academic studies may be so innovative, but lack of fully completed reporting. And also, regulators often expected to science is complete, especially for chemicals requires careful evaluation. It takes time, but regulators have no enough time. And also there is no standardized weight of evidence framework. And that's the reason different agencies and different countries use slightly different criteria. For example, it's a accuracy and the other criteria. And that's the reason every decision and risk being challenged legally or politically. Why did you prioritize this data, not select this data? They can make these kind of questions. And also there is a data gaps and also inconsistent data about exposure and real-time exposure. So that's the reason for new chemicals, productions, volume, and also we have no idea about their environmental fate of them. Even hazard data exists, exposure uncertainty makes the weight of evidence inconclusive and unreliable. Thank you.
Thank you, Professor Bensu. And now to Mr. Auger. We have a question also for you to continue the discussion you launched in your presentation related to sweeteners. So you highlighted this evolving dialogue between safety science, regulatory frameworks, and especially consumer perceptions around sweeteners. How can industry and regulators better bridge this gap between robust scientific evidence that you clearly presented in your presentation and public concerns that you also tackled in the presentation to build this trust, especially in today's highly polarized information environment?
Thank you for the question. Indeed, I would say the science is there, being done in control studies, observational studies. All those are helping fuel the discussion, the scientific discussion, and all are important. And I think even when we look at all the researchers' own comments and highlighting even the limits of their own studies, everything is robust. Our problem really comes from the translation of such results and the use made through the media and through maybe quick reactions by certain players to convey messages out. And sometimes I took the example of this cognitive decline paper making big headlines in many news outlets in September. If you look the actual paper, it's completely valid. And they highlight themselves the limitations, and they've checked exposure just once 8 years ago in a questionnaire, and that's it. Nevertheless, we still end up with explanations to do. So I think that's a very essential question and an ongoing effort by all players. How do we try to do better? And in our view, as one industry sector, just on our part on sweeteners, that would be around explaining further by providing materials, for example, showcasing the pyramid of evidence Dr. Bensu showed in her slide deck, explaining what is an ADI, what it's not as well, just trying to convey sometimes fundamental baselines, toxicology elements, some science literacy as well. I think that's one thing we can do, and trying to equip certain colleagues who are also talking to consumers. So it's not directly from, in our case, industry to consumers, but when I think about, especially in the nutrition world, because we are in this intersection on the sweetener case between also the toxicology, for example, and nutrition science, all the people in the nutrition world, dietitians, nutritionists, talking to patients, explaining them basic things on toxicology and how things were assessed. So that's part of this educational aspect, I would say, which is essential.
Thank you so much, and how careful we should be when we communicate. So that's an invitation for all of us to rely always on science and evidence when we formulate any kind of communication in this regard. Thank you so much. So I asked you to keep your questions for now, so we have time just for one comment or question, of course. So please, if we have a microphone for just one question, but again, I will kindly invite all our audience to engage with our speakers after this session. So bring your thoughts, your comments, your questions, and I guess I can speak on behalf of all my speakers. We are ready to answer all your comments and questions. Okay. So we have Dr. Moaz. So
good morning. I'm Moaz Sana from WHO. Thank you for the presentation, and you highlight the problem of communication. You agree about what is available as science, but at the same time, you bring some confusion. I'm sorry to say that. When you read the WHO guideline and recommendation saying WHO suggests that non-sugar sweetness not be used as a means of achieving weight control or reducing the risk of non-communicable diseases, it is a contextual recommendation. But if you need to have very objective communication, you need not to stop to the recommendation and provide the rationale, because there is a short rationale that you can copy and paste in your presentation, explaining why the WHO group decide to provide this recommendation. One of them, and it is based on the available study, random control study, and for the adults, it showed that if you are substituting sugar with non-sugar sweetness, you see low calorie, you reduce low calorie, and you observe loss of weight. But the majority of the random control study are for only 3 months. In addition to that, the few number of studies that last more than 3 months didn't show evidence of body control for more than 3 months. So adding a study, one single study outside of all the available study and say, oh, look, this study is showing something completely different, I think is not very constructive. And we need, I understand from your point of view, you are pro non-sugar sweetness, I understand you, but at the same time, in WHO, in JECFA, we have one very good study, the Nutri study, that shows a good statistical association with all the consideration of confounding factors. But we didn't take this study because there is only one single study showing the association, and we said there is no evidence, and we said we will not consider aspartame as carcinogenic because we don't have enough evidence. So you see, what you are doing is balanced, very balanced, both sides. But I recommend and urge you that when we try to communicate, we try to communicate in not just giving the title and try to provide the rationale. And another thing, in our communication, we spend a lot of time to show what IRAC classification is providing as information at what JECFA did. You need to understand that the class 2B is possible. Does it mean carcinogenic? And what is meaning? Meaning that there is uncertainty, and it is a call for researcher and for sponsor to collect more data to better consider the uncertainty. So it doesn't mean it's carcinogenic. And that's why we need to be more careful about the communication. Thank you.
Thank you, Dr. Moaz. And thank you for bringing also this other point of view and helping us also to build a more balanced, as you said, a communication in this regard. Thank you so much. And with that, because we used all the time allocated for our session. So here, first, let me extend my sincere thanks to our speakers. So please join me in a final round of applause for all our speakers. For Dr. Batul also for co-moderating this panel discussion, for all the participants that stayed with us. Today's session highlighted a clear message. We must anchor food safety decisions in science while understanding and responding to the emerging complexities in perception and policy. On behalf of the Global Food Regulatory Science Society, G-Force, and the organizers of the Dubai International Food Safety Conference, thank you for joining us today. We hope you continue to engage with us during the conference. Enjoy the rest of the sessions. Thank you so much.